Hi there. I was just listening to some Schubert, and I thought you might enjoy that to start off today's VerEvMed lecture. Hopefully, that was fun for you. It certainly is something that I enjoy doing. Listening to music while I prepare these lectures and while I do research for my clients at VerEvMed and while I work on my Authentic Biochemistry podcast is a very frequent event. And so I hope that you enjoyed that. I'm going to try to improve, obviously, the audio rather than just coming from my iPhone, and try to figure out how to hook it directly into this presentation, this video presentation. I'm sure I can figure it out with a little bit of help from my friends. So let's get started. What I want to do today is discuss as part 4 how the biochemical architecture of Alzheimer's disease can be understood at the level of biochemistry, molecular genetics, environment, and immunoepigenetics. And in order to do that, I had to go through the first 3 lectures, which included, of course, discussion of bioenergetics, a couple of new publications that have come out relative to ATP being a major component in neuronal bioenergetics, driving changes in fuel utilization in the neuron and switching from glucose to ketone bodies generated by a Sphingolipid degradation pathway, which destroys the myelin sheath, which is generated via the astrocyte-associated oligodendrocyte neuron system. The degradation of the myelin sheath generates ultimately ceramide, and ceramide is a lipid, sphingolipid, which actually induces apoptosis or programmed cell death. But along with all the nuances of that, production of that particular membrane lipid, we also had a switch from ATP production from glycolysis to ATP production from the utilization of ketone bodies. Those ketone bodies were generated in the astrocyte, translocated to the neuron, thus blocking via interactions at the blood-brain barrier, utilization and uptake of glucose. So having basically a hypoglycolytic system, which is pro-dromal in Alzheimer's disease. So we went through all that. We also discussed in great detail how that system comes together in terms of ATP synthesis and utilization, electron transport chain, how that's blocked, how reactive oxygen plays a role in enhancing the production of pro-inflammatory cytokines, which further leads down the very dark corridor, which is Alzheimer's disease in aging human beings. So today I'm going to talk about part 4. This is my most enjoyable probably because it's lipids. And as being a lipid biochemist, I really like to demonstrate how lipids play a major role in biology, and often in normal physiology, healthy physiology, lipids are very specific and wonderful. However, when there's a disruption or dyslipidemia, lipids can play a very dark role, very negative role in physiology, therefore generating a disease physiology or pathophysiology. That's what we're going to talk about today. So anyways, title of today's talk is Sphingolipid-mediated neurodegeneration and a preliminary diaeventontologic of Alzheimer's disease. Don't worry about that last part. If you've been following along my lectures, you know I've generated a new paradigm in understanding biosystems, and that is a discussion that involves dialectics, event ontology, and an ontological temporal perspective, which comes together to fuse a trigonal planar system, which includes genetics, the environment, and immunoepigenetics. So that's a couple of sentences of what I mean by a diaeventontologic of a system. And this system we're going to talk about today is Alzheimer's. That's me in Utah a couple of years ago, actually. That's my email address. You can contact me there. That's my direct email address. Give me at least a day to get back to you. You can talk to me about any of the video lectures you've seen here and make comments, suggestions, and even potentially become a client from VerEvMed. VerEvMed is a company that this is These video lectures are an offshoot of a company that myself and my other co-founder put together. What we do is we look at primary scientific literature, we explain it, the evidence that is found therein, and then we verify that evidence. That's why it's called VerEv, for the biomedical community. This includes physicians, physicians' assistants, nurses, and of course, lay people. And we also bring forth the topics in the published literature for students, medical students, graduate students, undergraduates, as well as veterinary students, and even the legal system, lawyers and attorneys who want to know something about the pharmacology of a system vis-a-vis related to maybe a lawsuit or a class action suit or something like that, so they can prepare briefs. So that's just a very brief summary of what we do in VerEvMed. I also just mentioned to you that I've just started. I've had 3 podcast presentations, what I call authentic biochemistry. You can find me on all of the platforms, including iTunes and Stitcher. And we hope to be able to get more involved in the podcast and get feedback from our audience so that we know what level we should be talking about. Right now, I'm gearing my podcast at the level of probably a person who has a college education and has had some chemistry and biology. And I'd like to keep it that way because I'd like this to be a teaching event. So what I really need is feedback from my audience, and I want to build that audience because I'd like to be able to make this podcast available to everyone. That is available in the sense that they gain something from it. It's already available and free, of course, because I'd really like to get the information that is in the primary scientific literature out there into the community, out there into the world, because it's unfortunate that a lot of what's discussed in science on network and on digital devices and through the media is terribly distorted from what's really going on in science. And that's because most of the people that are presenting it are not scientists, don't have any background at the bench, don't understand how to do experiments, and certainly don't spend much time reading the primary scientific literature. And that's what I do. So anyways, this is coming from VerEvMed Studios in beautiful Pacific Inland Northwest, somewhere out there. This is, again, my video lecture series. This is it started out in January 2019. It is now, this series started out, we talked about molecular neurobiology, a paper published in January 2019. And we've been going now for about 5 weeks into this series. It's number 4, like I said. So right now it's March 16th, 2019, to put a timestamp on it. That's my Facebook page right there. And you can find all my video lectures and a lot of other things about VerEvMed and a little bit about metaphysics and Western philosophy, and even some music at my Facebook page. That is it. It's my formal Facebook page for VerEvMed, but it has all those other things too. Again, let's get started. This is, of course, Utah. All right. So we talk about a paper published in Molecular Neurobiology, and what it says, basically summarizing the introduction, is that there are 2 widespread neurodegenerative diseases that we probably are all aware of: Alzheimer's disease, AD, and Parkinson's, which is PD. There are neuropsychiatric presentations of dementia, and it's documented in about 47 million people worldwide. And that's where AD is the most common. There's about 70% of the Alzheimer's disease that's actually diagnosed because of the presence of aggregates of pathologically misfolded proteins. Now we have a misfolded protein that causes a pathology. We call that proteinopathy or proteinopathy. Okay, so I'll be bringing that up again and again just so you know what that means. Now the 2 that you've heard of course, even if you don't follow science and you're not a medical professional, are the extracellular senile plaques, built mainly of the amyloid beta or A beta protein. And that's actually a product of proteolytic cleavage of a transmembrane A beta precursor protein, or A beta PP. And there's 2 enzymes required to break down that precursor protein to the A beta peptide. And they're called secretases because they're secreted from the cell. And they're beta and gamma secretase or secretase. Alzheimer's disease further presents, as most of you know as well, with a microtubule-associated tau protein complex. Tau is a cytoskeletal glycoprotein. It becomes hyperphosphorylated. When that happens, it aggregates, and when it aggregates, it can form intracellular neurofibrillary tangles. Now, both the plaque and the tangle are associated with Alzheimer's disease, but neither one of them is causative in any research that's ever been shown. It doesn't cause Alzheimer's disease. It's just correlated. In fact, it's not always correlated. It's just those are two paradigmatic diagnoses for having Alzheimer's disease, either one or both, the tangles or the plaques. This is, of course, a lot of discussion has been shown in autopsy, postmortem autopsy in the AD brain. So quickly with PD, because we're not going to talk about it except for right here, it's the most common movement disease, and second only to Alzheimer's disease and neurodegeneration. About 1% of people over the age of 60 and 4% over 85 contract that, so it's much less than AD. Pathology involves subcortical neurodegeneration, a loss of dopaminergic neurons in the substantia nigra, pars compacta. There's a loss of dopaminergic striatum innervation in Parkinson's disease, and there's a histopathological aberration in the form of a protein called alpha-synuclein, containing an intracellular Lewy body or LB, so you also get Lewy neurites. It's a particular kind of histology that has been described, histopathology. There are disturbances in neurotransmitter systems, including the serotonergic, the noradrenergic, and the cholinergic, and of course you get dementia also in Parkinson's like you do with AD. So like AD, PD is a neurodegenerative disease that progresses without any obvious external symptoms for a while. And by the time they appear, the dopaminergic neuronal degeneration is well on the way. So it's really one of those terrible diseases where you don't know until it's too late. Now, that's all I really wanted to say about Parkinson's. We can do another whole series on Parkinson's. Of course, we should sometime. Let's get back to the topic of today. That involves a discussion of Sphingolipids. So I gave several lectures late last year, 2018, and also all the way in the summer of 2018. So now talking 9 months ago, about lipid metabolism. You can go back and look at those lectures. They're basically just a lecture on lipids, what the structures are, what their function is, how they're found in biological systems. So structure-function relationships, typical biochemistry lecture material. So I'm not going to go back and talk about all that today, but you can go back and review that. And I think that might not be a bad idea, especially if you're a biomedical professional and you want to have more of the actual concrete discussion about what these lipids are. But today I'll do enough so you'll know. Anyways, several sphingosine-based bioactive lipids are found within membranes. They're also trafficking through them, and they have been implicated in both normal and pathophysiological states. They exist as regulatory signaling molecules. So like all lipids, they form a structure of a membrane. They can be stored. Now, glycerolipids are stored when used in bioenergetics, such as triacylglycerol, generated fatty acid. But even sphingolipids have fatty acids, of course. They just have a sphingosine base rather than a glycerol base. And the fatty acids associated with sphingolipids can also be involved in bioenergetics, which we talked about before. You can beta oxidase these fatty acids, generate acetyl-CoA. Acetyl-CoA can be used in ketogenesis to form acetyl-acetyl-CoA, then acetoacetate, and then beta-hydroxybutyrate, which can go into the bloodstream and can go completely homeostatic and become a carbon source. That's what happens during starvation as adipose tissue is being delimited. But also you get specific ketogenesis in different organs. Usually we think about the liver generating most of the ketone bodies or the muscle to some extent, but also in the rare event when you get ketogenesis in the central nervous system, that is one of the paradigms for pro-dromal AD I've been discussing. Okay, so that's important to know. Besides sphingomyelin, the major lipid found in the myelin sheath of critical neuronal tracts in the CNS, that's what sphingomyelin is. The associated metabolite of sphingomyelin is called ceramide, ceramide 1-phosphate, sphingosine, and sphingosine 1-phosphate are all necessary bioactive sphingolipids derived from sphingomyelin, which is a specific sphingolipid found in the central nervous system, and other places too, of course. So the role of cerebrosides, which are more complex sphingolipids than what simple sphingomyelin is, which is just basically ceramide with phosphatidylcholine. Cerebrosides, which include galactosyl, glucosyl, sialyl, and sulfonyl sphingolipids. We don't really have enough information yet about how cerebrosides may play a role in Alzheimer's disease. There are papers out there and we can talk about it. But right now, I'm really keying in on sphingomyelin, that more simple, otherwise still somewhat complex lipid motif and metabolic system network grid. Anyways, going back to just the sphingomyelin derived, those cellular and extracellular trafficking sphingomyelin sphingolipids are interconvertible via enzymatic pathways that are not always expressed constitutively in any cell, tissue type, or across time. So you can't simply say that a given sphingolipid plays a role in Alzheimer's disease. You have to see whether or not it's in this particular tissue type, cell type, and when it's occurring and how it's occurring. That is the rate of its synthesis, total accumulation, whether or not it's secreted, whether or not it is sequestered, whether or not it is metabolized, not quickly. So again, it's a diaeventontological system, right? It's something that's happening as an event ontology through time, right? Denominator is delta t, small t time. So these sphingolipids are implicated in various physiological phenomena, including inflammation, stress resistance, proliferation, differentiation, and in the plastic and elastic remodeling of neuronal and glial sulfates in the CNS. I don't want to go back in and go through a long discussion about plastic and elastic, but plastic phenomena are things that, by that I mean that you leave an impression behind after you've carried out a biochemical event, so it's long lasting. Whereas elastic means it can be immediately turned around and erased and go 180. Plastic and elastic systems happen all the time in biological systems. So anyway, there's that. These potentiating sphingolipids are implicated, again, what are they implicated in? Cell division, growth, apoptosis, autophagy, senescence, adhesion, migration, inflammation, angiogenesis, and very importantly, endosomal plus organellar intracellular trafficking. Yeah. So they can carry packages of these lipids outside the cell they were synthesized in and basically act as trafficking rafts. Opposing or reinforcing modes of action have been ascribed to individual sphingolipid classes and molecular specific subclasses. For example, ceramide is a harbinger of programmed cell death in the general sense, whereas ceramide 1-phosphate and sphingosine 1-phosphate sustain cell vitality and transformational viability, meaning they can actually induce the dreaded tumorigenesis. So you see, you're playing a game here. It's actually similar when you think about phosphatidylcholine versus diacylglycerol, or phosphatidic acid and diacylglycerol. But those are glycerol-linked lipids, and we're not talking about those today. So mere addition or removal of ATP derived anhydride phosphate as an ester to preform polypeptides can reverse the direction of signaling without any change in transcription or translation. We call those things kinase cascades. Kinase means you add anhydride phosphate. So change in transcription or translation doesn't happen of the participating molecules, thus providing a molecular circuitry network to mediate cell fate. So it does, I guess, also prove it in terms of it's causing it to occur. So it's proving and improving, but I meant really providing there. It is therefore not a surprise to biochemists that bioactive sphingolipids will be involved in the initial and the closing arguments toward aging and neurodegenerative disease. For my friends and family who are attorneys. All right, here's just the basic biosynthetic pathways. I told you I wasn't gonna, I wouldn't say the word bore because nothing about pathways is boring. Never say that, never think that. It's always interesting and fascinating, especially when you see structures. But basically, you synthesize sphingolipids starting from palmitoyl-CoA and serine. In a pyridoxal phosphate mediated manganese mediated reaction, you make 3-keto-dihydrosphingosine, which is then, of course, reduced to sphinganine, or also known as dihydrosphingosine. This molecule here. So basically, you're taking something out of amino acid pool and something out of the fatty acid pool, and you're starting to generate a sphingolipid backbone. So ceramide biosis is controlled by that serine palmitoyl phosphotransferase. That's the first reaction there, also known as SPT, whose product is the dihydrosphingosine. It's converted to by ceramide synthase to dihydroceramide, which is then converted to dihydroceramide desaturase, that enzyme, DES or DEGS, to ceramide. Okay, so this is showing you this pathway here. There is serine and palmitoyl-CoA. You go through this 3-keto-sphingosine, dihydrosphingosine, or sphingonine, right? And then ultimately you make dihydroceramide, and dihydroceramide goes through a desaturase reaction, leaving a trans double bond actually, making ceramide. Okay, so it's on the way to making sphingomyelin, but ceramide is the central player here that we're going to discuss. So ceramide can be a precursor to axonal localized oligodendrocyte generated sphingomyelin. So axonal from the neuron, localized oligodendrocyte generated, that's where the lipid comes from, sphingomyelin, by the sphingomyelin synthase systems, while the number of inducible and constitutive sphingomyelinases may resynthesize ceramides, and that's the downward trend here. These are just drugs that have been described to block various components of the sphingolipid canonical pathway. Don't worry about them now. We talked about them before. We'll talk about them again for sure, but not today. Ceramide also then serves as a precursor to the production of the Sphingosine, see here, by an enzyme called ceramidase. Okay, so Sphingosine then can be phosphorylated, sphingosine 1-phosphate, and then that can be completely broken down to ethanolamine 1-phosphate and palmitaldehyde. And these have no biological activity that we know about yet. It doesn't seem they can be used as precursors either, because you need serine and palmitoyl-CoA to drive the de novo biosynthetic pathway. So that's really important. Ceramide is a precursor to these other really important lipids. Now, what I'm not showing you here, I don't see it, is ceramide phosphate. And there's just a kinase, which will take the lipids ceramide and add a phosphate to it. So we talked about already. You got ceramide 1-phosphate, sphingosine 1-phosphate. Very important 180-degree differential in how they mediate cellular responses than the parent molecule, ceramide. So ceramide serves as a precursor. So these are the kinases we just talked about. Some of these are inducible. We'll see soon. Highly regulated fashion in various cellular compartments. Dephosphorylation is carried out by phosphatases. Okay, so you can remove the phosphate. That's the backward reaction there. While sphingosine 1-phosphate can be hydrolyzed irreversibly, as I just told you, with this lyase reaction to these biologically inert products. Now, there's a ceramide to sphingosine pathway. There's a ceramidase. There's a ceramide synthase pathway we just looked at. There's the kinase, ceramide kinase, going to CYP, and then it's binding, we think, to some receptor. Now, sphingosine, and we know the ceramide also functions at some level like that, but then we have sphingosine 1-phosphate, and there are multiple receptors for this sphingosine 1-phosphate. Receptor 2, 3, 1 to 5, or 2 to 5, working through different G-coupled protein complexes, inhibitory and activating. They work through then a secondary signaling system, either phospholipase C, and with the production or that is the induction of calcium uptake, or that is released into the cytoplasm, and that then can turn on protein kinase C and nitric oxide synthase, or the inhibitory protein can work through phosphoinositol 1,3 kinase, or the ERK kinase, and AKT, and that can all block the FOXO, GSK3 beta, and BAX-BAD pathways. Some of these in particular are associated with apoptosis. And then there is the receptors 2 through 5 here, which they can work through G protein 12-13. We talked about that recently, which works through the Rho kinase pathway, and that can induce the NF-kappa B, and that can then generate or activate the inducible nitric oxide synthase. So you see all these secondary and tertiary signaling molecules are linked to the ceramide pathway. It's really important for you to understand that. Very, very potent compounds. So the plasma membrane is the structural domain for the sphingosine kinase reaction. Sphingosine 1-phosphate and probably ceramide 1-phosphate can bind cell surface receptors. That's where it's localized. Those receptors belong to the EDG family and bind to those various G protein complexes and Rho proteins. We just went through how they're functioning. Sphingolipids also modulate signaling events such as cyclic AMP production, MAP kinases, which of course is mitogen activated protein kinase C, phospholipase D, and the PI3 kinase AKT PKP pathway you see up there. It's the phosphatidylinositol 1,3 kinase AKT pathway that co-distributes metabolic and stress signals engaged in energy metabolism through aging to age-related diseases that I've talked about already. So I know you would like it if I talked about it again, but I'm not today. Now, sphingosine 1-phosphate through its G protein coupled receptor regulates oxidative and nitrositive, that involves nitric oxide, stress and death survival. Okay, so it can act as a rheostat, that's what they call it, right? You can either crank it up on one side or the other. It's like a teeter-totter in a way, right? Or balancing act. So as sphingosine 1-phosphate receptor 2 inhibits AKT, while the receptor 3 activates it, inducible nitric oxide synthase is typically induced by NF-kappa B, which transcription factor as well, which can also undergo sphingosine 1-phosphate, ceramide 1-phosphate dependent suppression of expression via P38 MAP kinase. Ceramide thus participates in the control of senescence, differentiation, and neuronal arborization, something I talked about in a previous VerEvMed lecture. Hopefully you caught it. It was when I was talking about actually drugs. While free sphingosine may also modulate cell death, and ceramide 1-phosphate stimulates cellular survival. And they may counteract ceramide itself signaling by decreasing acid sphingomyelinase activity, which remember generates ceramide, and serine palmitoyltransferase activity, which is the rate-limiting step for the de novo synthesis. So sphingosine 1-phosphate regulates cell viability, neuronal excitability, and arborization. Sphingolipids are also engaged in immune phenomena, which critically alter the fate of brain cells, such as with microglial activation in glioblastoma, one disease we've probably all heard of and some of us know very intimately, and other neurodegenerative disorders, such as we're talking about today, Alzheimer's disease, Parkinson's disease, Lewy body dementia, prefrontal dementia, things like that. The roles of sphingosine 1-phosphate and ceramide in the survival of brain neurons are far more complex than the antagonism described in the sphingolipid rheostat model you see here. Okay, that's what I'm going to tell you today. So they're just talking about, they call it rheostat. They say if ceramide's up, sphingosine 1-phosphate down, you get AD. If ceramide's up and sphingosine 1-phosphate is variable, you might get PD. And if ceramide's up and sphingosine 1-phosphate's down, similar to here, you get just normal aging being enhanced, right? So aging is linked to Alzheimer's disease. So maybe this is how it's linked, right? By this change in membrane lipids, which changes everything about cell fate and cell-to-cell interactions, including with the immune system. And if you watched any of my lectures on aging, you know that's the case because we dig deeply into the literature on that. Okay, so these things here, these two diagrams, A and B, one is showing you when you have high levels of ceramide versus high levels of sphingosine 1-phosphate. So S1P and ceramide antagonistically signal cell survival or death being by ceramide, largely via shared mediators, and both can directly inhibit each other's synthesis. Wow, so it makes it already one level more complicated than you might want it to be if you wanted to target this pharmacologically. So sphingosine 1-phosphate is classically viewed as anti-apoptotic, that is, that it prevents cell death, programmed cell death, and has been shown to mediate the actions of numerous anti-apoptotic compounds. S1P typically opposes the pro-apoptotic role of ceramide, presumably by decreasing oxidative stress and modulating the expressions of pro- and anti-apoptotic proteins of the BCL2 family. So modulates the pro-apoptotic proteins by knocking them down, pushing them down, suppressing them, and it activates the anti-apoptotic proteins in the BCL1 families, therefore depressing or decreasing programmed cell death. So you can take a look at all these different proteins involved here, but basically here's ceramide being synthesized by a pyrophosphatase, removing that phosphate from the ceramide. You've got all these early in the pathway enzymes, ceramide synthase. the serine palmitoyltransferase, the desaturase enzyme. You make ceramide. It activates a phosphatase, which then blocks AKT. When AKT is blocked, it won't block this. So if that's not blocked, it can induce this whole pathway here. You build up ROS, you get cytochrome C release, you get all the caspases. I think you know where I'm leading here. This is programmed cell death via the mitochondrial system. Beclin-1 and LC3-Roman numeral 2 are generated. You get axonal degeneration and neuronal death. Ceramide also seems to induce reactive oxygen species, and that blocks the sphingosine kinase, which would otherwise pull that lipid into sphingosine 1-phosphate, thus inhibiting this whole system, draining this pathway. Anyways, what ROS does is turn on the junk P38 ERK pathways. That works through stress phenomena, cell cycle stop systems, P53, AP1, and all that basically is to program cell death. Kill the cell, there must be something wrong with it. Kind of the exact opposite happens here, sphingosine 1-phosphate. It inhibits the sphingomyelinase, so you don't make ceramide. The sphingosine kinases actually are one of the reasons it's made. If you have high reactive oxygen, that's going to generate P53, and we just showed you over here, P53 can actually block the synthesis of sphingosine 1-phosphate. That's a signal that says kill the cell, don't mess around with trying to retailor it or autophage it out. Moderate reactive oxygen actually can induce sphingosine kinase, that's cell survival. It works through TRAF2, through an NF-kappa B pathway, directly inhibiting neuronal death through that system. It can also work through these receptors we talked about, again through the same type of protein, junk P38 and ERK. These are all like canonical actually in a lot of immune cells and endothelial cells, inducing AP1, okay? And then here with this pathway, because you have all these other proteins altered, they're flipped in terms of their reactivity, and they're downstream from these other effector distributed network systems, you get inhibition of neuronal death. So there's all the abbreviations. So sphingolipid signaling controls transcription factors AP1 and NF-kappa B. Those are two major transcription factors in the cell, and those basically control cell death and inflammatory responses. AKT targets the FOXO1A, 3A, and 4, and 6. They're all engaged in cell death regulation in human tissues. Okay, so you see where that links in. The prevailing role of elevated ceramide in cell degeneration, death, is mediated by multiple signals, as we just saw: inhibition of mitochondrial respiration, generation of reactive oxygen, blocking of NADH oxidation, for example. Partially reduced forms of oxygen will do that to you. Inhibit complex 1 in the electrotransport chain, that is. BCL2 binding protein, Beclin-1, and autophagosomal LC3-2 Roman numeral, which binds mitochondrial ceramide to induce lethal mitophagy that start to cause the degradation of mitochondria, which of course can induce programmed cell death. While inhibition of the HDAC, that's histone deacetylase 1 and 2, is engaged in the pro-survival signaling of sphingosine 1-phosphate. So you're inhibiting the deacetylase, which means you're leaving histones acetylated, which means you're enhancing transcription and therefore protein synthesis down the road, leading to an entire legion of proteins that are there to repair, recombine, and otherwise fix all the macromolecular structures so that the cell survives. Autophagy can also be turned on depending on the bioenergetics of the system and the cell type. Okay, so pro-survival signaling through sphingosine 1-phosphate activates those enzymes P38, ERK, and it can block the junk pathway. ERK appears to mediate the pro-survival action of sphingosine 1-phosphate via the receptors. They also stimulate, as we just saw, the anti-apoptotic P13-AKT pathway, whose disruption in AD may deploy a disease pathomechanism. So we disrupt that. We already know that's pro-dromal for AD. The sphingosine 1-phosphate, S1P, can also exert neurotoxic influence, however, depending on the spatiotemporal control of its production and degradation, or when its concentration reaches too high. Loss of physiological ceramide concentrations can also lead to structural disturbances in the mitochondria, and it can reduce respiration. Ceramides also regulate membrane fluid dynamics, thus via cholesterol mediation in some membranes, like the plasma membrane, thus influencing other aspects of organellar function and movement and tropism, and of course life cycles such as mitochondrial fusion, fission, and vesicular transport. Life cycle there of the organelle. The peroxisome is a similar life history, by the way. So their pathophysiological roles are dependent on their chain length and the ceramide synthase isoforms that make them. Okay. Ceramides lead to dephosphorylation and inactivation of AKT in the general, with the bad and glycogen kinase 3 beta active, glycogen synthase kinase 3 beta activation. That's all the transcription factor stuff. Don't worry about the fact that it's got a glycogen moniker to it. It's because it was first discovered there, and it actually does work on glycogen synthase kinase. Those changes, though, in general, all happening in the central nervous system, causes mitochondrial alterations and via cytochrome C release and the activities of those caspases we saw on the previous slide, all associated with axonal degeneration and death of the neuron, all pro-dromal and continuing on through the median and late stages of Alzheimer's disease. Ceramides might also directly form pores in the outer mitochondrial membrane. Not a good thing because it's going to uncouple electron transport, for one thing, and block ATP transport. That leads to the release of cytochrome C2 on the first breath and blush, and other proteins inducing, of course, programmed cell death, the mitochondrial type. Ceramide-induced apoptosis thus involves both caspase-mediated and caspase-independent pathways. We talked about that before in other talks. Cumulative balance between the S1P and ceramide will influence apoptosis and, of course, cell survival. Change the regulation of autophagy by the complex interplay between mTOR, Beclin, and BCL2. S1P-dependent autophagy is said to be a homeostatic mechanism, pro-survival response involved in the clearance of intracellular debris, which can happen after an inflammatory response. So, I've seen this slide many times if you've been following all my lectures. Here's sphingosine versus ceramide. There are the enzymes that kind of keep that rheostat open. Remember, you can phosphorylate sphingosine, you can phosphorylate ceramide. CYP is about cell survival, proliferation, and inflammation, as is sphingosine 1-phosphate. So I guess that's KIP and CYP. That also, sphingosine 1-phosphate is also going to involve in cell survival, proliferation, inflammation, but also cell motility, as we just mentioned. Of course, it could be degraded out via this pathway we already saw. And here are the de novo pathways, and here's it coming from sphingomyelin. And we know that ceramide turns on apoptosis, cell cycle arrest, and cellulosenescence. And in fact, the parent sphingosine base also does apoptosis and cell cycle arrest via the P53, P38, AP1 pathway. Corruption thereof. All this is from a paper journal of Cell Science way back in 2005. That is this slide. As I discussed recently, ceramide plays a significant role in prodromal AD by linking A-beta-induced sphingomyelinase activity, generating demyelination with a biofuel substitution from glucose, sorry, it's not a space there, to ketone bodies in astrocyte-associated adjacent neurons. That one bullet point there was the topic of the first 2 full 1-hour-long lectures in this 4-part series so far. Other evidence suggests that additional sphingolipids may be involved. The canonical, if there is such an adjective for AD, the canonical mechanism in AD involves neuronal and synaptic degeneration in AD's central nervous system that follows a pattern of ROS accumulation and damage. Damaging influences then alters the electron transport chain. It alters peroxisomal redox signaling, which alters mitochondrial and peroxisomal dysfunction, that is, it enhances it. Then you get early phagy, mitophagy, and peroxiphagy. That causes glucose hypometabolism, so decrease in utilization and the uptake of glucose. GLUT4 doesn't pick up the glucose in the astrocyte, for example. Then you go switch to beta oxidation of fatty acids, with the acetyl-CoAs converted to ketone bodies. You get ketogenesis. Then you get ATP depletion because of this, because ATP can't leave the mitochondria. It's not being generated by the pathway because you're ketogenic. You get calcium deregulation. Ultimately, you can get cytokine-mediated pro-inflammatory responses and neuronal death, all mediated from an astrocyte or even microglia or even oligodendrocytes associated with neurons. Many of those pathways are triggered and propagated due to the actions of soluble oligomers of A-beta protein on neurons and glia, and the role for ceramide S1P is coming into focus. That's what we're talking about right now. Lipid rafts also play a role here. They are cholesterol and sphingolipid-enriched microdomains of dissociated, usually plasma membrane, that functioning as signaling platforms. So they go around as a raft carrying a lot of this activity by bringing everything with them on the raft. All the lipids, all the precursors, and all the cofactors, for example. These rafts strongly associate with A-beta production, and both the beta and the gamma secretases are actually co-localized on that raft. The lipid rafts can mediate A beta aggregation into oligomers, and the A beta 42 oligomer such thing induces local membrane fluidic dynamic corruption, which ultimately can lead to neuronal death. So here's Sphingolipid metabolism in AD, kind of put together in a model. This comes from a paper, 2015. Sphingolipid metabolism correlates with cerebrospinal fluid, beta amyloid levels, etc. ER, you get a pro-acid sphingomyelinase, which acts on the Golgi. The Golgi then secretes an acid sphingomyelinase along with the lipid themselves. You can have two different types of endosomes. You can have endolysosomes and exosomes. Exosomes can actually leave the cell, pushing out this secreted acid sphingomyelinase, which again, these rafts can then do their own business. They show up then in the blood as nano or micro-sized particles, which carry with them all of the mechanisms necessary to carry with them this induction of the production of ceramide, which I just said is programmed cell death, as well as can cause neuronal injury, neuronal degeneration. So pro-acid sphingomyelinase is distributed between the Golgi secretory pathway and the lysosomal pathway, as I showed you. The sphingomyelinase, ceramidase, and phospholipases can induce the formation of secretory vesicles, because these are enzymes that actually can carve up the membrane lipids, creating the right biophysics so the vesicles are generated. Okay, so that's pretty cool, right? No fresh transcription here at all. You also get a blebbing of the plasma membranes to generate nano and micro-sized particles that leave the cell. And they have all different, depending on the size of the microparticles, sphingolipid composition. You also get then the secreted sphingomyelinases play a role in endocytosis, membrane repair, or remodeling. So they're altering membrane fluid dynamics by changing membrane fluidity via the enzymes that are associated with these particles functioning now on the outer leaflet of the membrane. Right, so they're altering the membrane. So the membrane is going to be all moving in one direction or another. That's basically what's going on here. Setting the stage so the cell will generate a neurodegenerative pathway. This is Alzheimer's disease. So you get this endocytosis. The letter E then shows you lysosomal acid sphingomyelinase, the L, plays a role in the fusion of phagophores with lysosomes. I talked about this a couple of lectures ago, resulting in autophagocytosis. Any dysfunction in that pathway, such as a decrease in secretory acid sphingomyelinase in AD, will result in changes in physical properties of the cell membrane. Okay. Anomaly in exocytosis, endocytosis, and defective trafficking of all these lipids and membrane-anchored proteins that go along with these endosomal raft complexes, resulting in a dysfunctional membrane repair and an altered A-beta excretion pathway. All of this is what happens to sphingolipid metabolism in prodromal early stages of Alzheimer's disease, way before symptoms show up. Okay. A lot of things going on here. So I talked about the sphingolipid rheostat. You got sphingosine kinase making the sphingosine 1-phosphate. That induces all the pro-growth pathways, angiogenic growth, proliferation. Also induces some inflammation and cell survival and angiogenesis, laying down a new blood vessel. Sphingosine itself is apoptotic, as is its parent compound, ceramide. You make it just by this ceramidase reaction here. Ceramide synthase will regenerate ceramide. So you can just see the structures are different. You've lost this fatty acid here. When you make sphingosine, that was the fatty acid you added. You had an N-linked fatty acid when you make ceramide. Of course, ceramide and sphingosine are both involved in apoptosis, but ceramide even also enhances age-related senescence and even cellular terminal differentiation. What turns on the sphingosine kinase are things like corticosteroids and sex hormones like estradiol, the HIF, the hypoxia-inducible factor, the ERK kinase, and even growth factors can all turn this on, making the more benign lipid causing cell survival. And all that's then described here. Several upstream activators, as I just mentioned to you, been shown to contribute to carcinogenesis. So if you make too much sphingosine 1-phosphate, you can get cell proliferation. So this is bad. That's why it's a rheostat. This is all the way in this direction, not good tumorigenesis. All the way in this direction, not good degeneration. Right? That's what we're talking about here. Now, Cellular rafts, okay, which we're dealing with here, you have an amylogenic and you have a non-amylogenic pathway, and you have an amylogenic pathway. Two different pathways utilizing various components of the A beta protein complex, along with the secretases, like the base secretase. There's a gamma secretase. This comes from a paper published 5 years ago now. Cellular rafts are mobilized by sphingolipid biophysical phenomena, thus correcting for and against tropism towards cellular membranes and docking receptor proteins with cargo ligands. That's what's going on here. Okay. Two different pathways. Cargo ligands are being presented to the membrane surface. Some of this precursor protein is being secreted here. If it's secreted along with activation of the gamma secretase, you get, of course, the A beta protein. Right? You also see this P3 protein is generated here. So beta cleavage of the A beta precursor protein. Ceramide linked rafts serve to stabilize the base enzyme. The beta site of the A beta precursor protein cleavage enzyme 1 is a beta secretase. That's what that is. Indeed, synthetic ceramide analogs in rodents modulate the gamma secretase, thus increasing the A beta 42 isomer with concomitant hippocampal and neuronal damage and resulting learning and memory deficits. So that's all because of ceramide, right? So synthetic ceramide analogs in rodents will modulate those pathways and induce in this Alzheimer's model, animal model. Positive correlation between sphingosine 1-phosphate production by the kinase and the amyloid beta precursor protein processing, where sphingosine 1-phosphate produced by the kinase 2 isoform may activate base 1, thus leading to higher release of the A beta peptides. Sphingosine 1-phosphate was shown to specifically bind to full-length base 1 to increase its proteolytic activity. So there's a lot of things going on here. Membrane fluid dynamics are changing. You're getting the secretion and production of specific isoforms of the precursor protein, and these can be functioned on different ways by the secretase enzymes, which of course are initially membrane localized, and they can produce the A beta peptide, and that A beta peptide is going to be bad news. That's normally, this is the bad pathway, the amyloidogenic pathway, because it involves then the oligomers. The oligomers induce all that other activity we just talked about. So, in addition, now this is really interesting. This came from a paper published in BBA, looking at hydroxylated docosahexaenoic acid, an omega-3 fatty acid that some people suggest is good in the diet. Check out what this paper suggests. An addition of a specific hydroxylated omega-3 dihydroxy docosahexaenoic acid, 22 carbon 6 double bond fatty acid coming off of the chain elongation, chain desaturation pathways from the omega-3 series parent compound, alpha-linolenic acid. From that pathway, you make DHA and then you hydroxylate it. That via an NADH-dependent enzyme actually can reverse sphingolipid-mediated AD phenotypes. That's what they're suggested here. So here you've got oligomeric A beta peptides, high affinity binding back to the membrane. This is an Alzheimer's disease neuronal membrane here. You get NMDA-mediated calcium exocytotoxicity in these membranes. This increases the A beta levels induced by stimulating the amyloidogenic pathway. Sphingomyelin and cholesterol-enriched liquid ordered domains are involved in this process. Lipid raft membrane receptors mediate tau hyperphosphorylation, now from the A beta to the tau fibrillary tangles. That induces neurotoxicity and ultimately neuronal death. All of this then is what you don't want to have. This paper suggested that if you add as a treatment animal model the hydroxylated docosahexaenoic acid, this alters membrane fluid dynamics. The oligomeric A beta peptides then are not associating with the membrane, low affinity binding because of the change in fatty acid composition. The NMDA-mediated calcium exocytoticity is blocked because of this change in membrane fluid dynamics, altering the way the membrane functions and what's in it. You get a non-association of the beta and gamma secretases. That's no longer there. So those lipid rafts aren't made. It gets a reduction of the A beta level because you don't have the secretases which cleave the precursor protein, blocking neuronal death. You also get a liquid disordered structure, and that whole thing tends to lead to this anti-prodromal AD pathway. So membrane receptor exit from lipid rafts reduces tau hyperphosphorylation. Don't get that anymore, and therefore no neurotoxicity from the neurofibrillary tangles. All of that then believing to be neuroprotective. And so this needs to be followed up in the animal model. It's an interesting paper, and it has been. Now there's more diaeventontological phenomena associated with AD. A mobilization of the Sphingosine kinase 2 from a cytosol to the nucleus is associated with A beta deposition. So just moving the kinase from the cytoplasm to the nucleus changes things. Therefore, the A beta production correlates with low Sphingosine kinase, that's when it's shuttled to the nucleus, for example, and high Sphingosine 1-phosphate lyase, because that generates back ceramide, as enabled by changes in subcellular distribution of those enzymes, as shown here. This is, again, reminding you of the pathway. Just so you remember, here's the serine palmitoyl-CoA transferase reaction, right? These are the addition of the fatty acid, ceramide synthase reaction, the desaturation pathway, make the trans-double bond, making ceramide. Ceramide transferase is transferred then from the ER. That lipid is transferred from the ER to the Golgi. Ceramide can then be acted upon, making the glycosphingolipids I talked about. That's a whole other pathway, really important in the central nervous system, or it can go down the sphingomyelin pathway. This is all normal physiology. Sphingomyelin, though, can also be degraded directly to ceramide. Ceramide can be phosphorylated. That's considered also a positive lipid, like sphingosine 1-phosphate. So you can see here how these three things are linked. Okay, this comes from a paper published in Journal of Lipids, 2013. Now, here is the nuts and bolts what we're talking about here for today. So you have sphingolipid levels changing, you have protein levels changing, you have activity of enzymes being altered, and you have transcription changing, all in the Alzheimer's disease pro-dromal paradigmatic system. So human AD hippocampus, sphingosine 1-phosphate levels, messenger RNAs for the SERK, that's the ceramide kinase, the sphingosine 1-phosphate receptor 1, sphingosine kinase, sphingosine kinase 1 and 2, all of those are reduced, okay, in the human AD hippocampus. That's not a good thing, you see, because you're generating more ceramide. Lower S1P levels, less receptor for it, and sphingosine kinase protein activities were associated along with elevated S1P lyase, removing that pro-active positive lipid, decreasing the AD pathophysiology and histology, and the hippocampal cortex, increased ceramide and sphingomyelin levels, mRNAs for ceramide synthesis, the acid sphingomyelinase, which of course makes ceramide, and sphingomyelinase protein itself, so you get transcription and you also get more protein made, so translational phenomena are all contributing to this. We're also observing the brain cortex. So you can see here in AD, you get the decrease in receptors for the sphingosine-based lipids, and you get a decrease in some ceramide synthase isoforms, which actually are co-regulated, which would tend to decrease or deplete ceramide levels in the long run. But you get a lot of ceramide synthesis 1 and 2 buildup at the transcriptional level. You also get the initial enzyme, the palmitoyl transferase enzyme. So this is all working in a very negative way in terms of transcription. I already told you the sphingolipid pathway a little bit, but ceramide with a C18 fatty acid is increased. Saturated fatty acid, ceramide with a C24 fatty acid is increased, where sphingomyelin with that lipid can go either way. Sphingomyelin also can increase the amount of stearic acid, as with the ceramide, and you get a decrease overall in the lipid levels of sphingosine 1-phosphate. Protein level follows, kind of mimics messenger RNA, transcript to translate to protein. You see some of these receptors going down, block the sphingosine pathway. You get increases in the sphingomyelinase activity, as well as the protein levels. So this is all negative and all why we talked about before. So the A beta protein also disturbs sphingosine 1-phosphate signaling, potentially shifting the balance towards much more pro-apoptotic state. The sphingosine kinases regulate the severity of inflammation associated with the ApoE, another protein associated with Alzheimer's disease, ApoE4 actually. where there is a 2.5 times higher sphingosine 1-phosphate to sphingosine ratio in the hippocampus of APE2. That's a healthy apolipoprotein off the HDL fraction versus the AD-linked ApoE4 isomer. So there are published reports on ceramide-dependent death of oligodendrocytes induced by the A-beta via activation of neutral sphingomyelinase, while the suppression of ceraminidase activity is positively correlated with increases in the A-beta protein. Other work has been demonstrated to show that the A-beta induced sphingosine kinase inhibition with concomitant ceramide accumulation, where cell viability was restored via expression of a sphingosine kinase. So in other words, when you get that system, if you add back a plasmid that allow you to express sphingosine kinase, outside of the nucleus, or get it introduced back into the nucleus, then you correct all this problem that's caused by the ceramide pathway. In a glial cell line treated with A-beta and with lipopolysaccharide and also with interferon gamma, pro-inflammatory stuff here, an increase in the inducible nitric oxide synthase associated nitric oxide production increased ceramide accumulation via that then induced an activation of the neutral sphingomyelinase, and that was similar to events using oligodendrocytes treated with the A-beta protein alone or with, in combination, the pro-inflammatory cytokine tumor necrosis factor 1-alpha. So this comes from a paper published in Frontiers in Endocrinology. Again, sphingolipid-linked AD occurs within the multivariant pro-dromal disease environment. Now check this out. Now we're not talking about AD, we're talking about other diseases which have also been shown to be impacted by sphingolipids. So this pathway here, making sphingosine lipids, is known to be blocked in atherosclerosis and insulin resistance and non-alcoholic fatty liver disease. That's blocked when you have those diseases because of metabolites generated in dyslipidemia in those systems. But if you do make it down to sphingonine, ceramide synthase is still functional in Alzheimer's disease, for example, making dihydroceramide. But once you get to the desaturase step, again, things like type 2 diabetes, obesity-linked diseases like insulin resistance, endothelial dysfunction like in cardiovascular disease, that all blocks the synthesis of ceramide. Remember, ceramide is a programmed death signal. So, for example, if you get cardiac hypertrophy, which is associated with obesity, you want to be able to remove, you don't want a lot of fibrogenesis in the hearts for the hypertrophy, and ceramide would block that or remove those cells. Here you're getting a blocking of the synthesis of ceramide in those diseases. Okay. Likewise, insulin resistance and atherosclerosis will prevent the utilization of ceramide to make sphingomyelin. Again, this could be in other tissues, but it's also in the brain. So that means that finding a drug that will tune up, say, one pathway or block one pathway may actually cause more problems if you've got a person that has a lot of disease orientations. And in fact, as I mentioned at the very end, obese people, people with type 2 diabetes, people with cardiovascular disease, people with early forms of various types of cancers, kidney diseases, liver diseases, muscle type diseases. It's all associated sometimes with a behavioral living pattern such as overeating or the wrong kind of food, pro-sedentary lifestyle, can actually all fight on that same pathway, but with largely distinctive characteristic changes in the vectorial transfer of going either from sphingomyelin or ceramide or sphingosine 1-phosphate. It's really important to keep in mind. You can't target these pathways with pharmacological drugs, pharmacotherapies, without also impacting a pathway which might actually induce or enhance diseases in other systems. So here's my slide showing you how I put together the AD Diaeventontologic. I want to show you here's Alzheimer's disease in the center. You have a genomic, an environmental, and an immunoepigenomic contributory trigonal planar system at work. I also want you to keep in mind something about formal logic, right? Okay, so the square of opposition and logic. Living systems interact according to a pseudo three-dimensional biological trigonal plane of opposition. What is that opposition I'm talking about in Diaeventontological paradigmatic systems? You can have the universal affirmative, for example, in AD, no harm to the individual, maximum benefit, rare or occasional epistasis, that means gene-gene or system-system interactions. You can have universal negative, severe harm to the individual, all the way full-blown Alzheimer's neurodegenerative disease. And you get 100% gene-gene interaction, 100% epistasis. You can get particular affirmative, going down the subalternate route in the square of opposition, some benefit to the individual, and a disinterested epistasis. In other words, it doesn't seem like it's coordinated to induce the disease. So some of it, so the person doesn't necessarily ever contract the disease, they die of something else. Or you can have a particular negation, and that's where some harm to the individual, and you get some kind of predicated epistasis, which can lead to increases in disease severity as you go from 65 to 70, 75 to 80, until finally full-blown Alzheimer's to death, which could limit the life of the person. So in the environment, what are we talking about in Alzheimer's? Well, first let's go to the genome. So you have to have, of course, the amyloid precursor protein. Okay, we all make that, but the abundance of it and the availability of it. You need to have the activity of the neutral sphingomyelinase and, of course, the acid sphingomyelinase. It has to be secreted. So that gene has to be there, and it has to be a certain particular single nucleotide polymorphisms of those genes are more prone to activation than others, as you might guess. That's a genomic phenomena. Ceramide synthase has to be there. That gene has to be expressed. The beta and the gamma secretases have to be made. Presenilin, some protein we didn't talk about today. The tau protein, ApoE4, the apolipoprotein E4 we just talked about. Other things that are genomically associated are IQ. Lower IQ tends to sometimes link to more severe onset of Alzheimer's disease, shown in a publication. Adipose equipose, so a person that has a lot of adipose tissue, particularly in the viscera, can be more prone to leading to a prodromal AD disease. And gender is a question mark. We don't know. Some of this early prodromal stuff with ceramide seems to be happening more often in women than men, but that really needs to be worked out. But there is a gender distinction in Alzheimer's disease. Men tend to get it more frequently a little bit, and they tend to get the full-blown disease earlier. So those are some genomic characterizations. How about the environment? Aging itself, inflammation, cardiovascular disease, any infections, viral, bacterial, fungal even parasitic, any stress to the system, which can include neuropsychiatric, which can all generate reactive oxygen. Sedentary lifestyle, which is going to alter that adipose equipose, but that's an environmental effect there, superimposed upon a genetic predisposition to putting on a lot of obesity. Obesity itself is environmental. That is, that people don't need to get obese or they can lose weight. It's an environmental effect. Education seems to play a role. More highly educated people tend to have a less pronounced AD, histopathology, pathophysiology, and also the ultimate symptoms that show neurodegeneration, such as loss of memory, loss of self, and ultimately full-blown dementia and death. So higher education seems to work against that. We don't really know what that is, but it probably has something to do with neural circuitry. Intellectual avoidance, people that keep their mind active tend to be able to stave off Alzheimer's disease or have it altogether not occur in them, even though other members of their family might. That's a genetic component, of course. Alcohol and drugs, that more excessive use of alcohol throughout life, more excessive use of recreational or even pharmaceutical drugs, which alter the central nervous system or peripheral nervous system, can also be lead to pathways that enhance the Alzheimer's disease diaeventontologic. Then finally, the immunoepigenome, which we're going to finally end up talking about soon, microglial activation, astrocyte dysfunction we just talked about, oligodendrocyte malfunction we just talked about. We didn't talk about beta and plasma cell recruitment, but we're going to. T-cell differentiation and modulation, we're going to eventually get to that. But we did discuss, or at least mention, cytokine, chemokine, growth factor modulation of this whole pathway. This is all part of the immunoepigenome, because a lot of this is associated with changes in splice frequency, changes in alteration of gene expression, not in the gene themselves, and these various cell types, some of which are immune cells. And then, of course, frank epigenetic phenomena, microRNAs are involved. We talked already about sirtuins, the deacetylases. We also mentioned in earlier discussions the histone acetyltransferases. Then there are the DNA methyltransferases and the DNA demethylases. All of those systems, those enzymatic systems, altering acetylation pattern, methylation pattern, as well as the secretion and utilization of microRNAs that can block certain transcription of certain genes. All of that epigenetics is linked up to the immunobiology of the system, which interacts in this three-dimensional state to get this Alzheimer's disease. Okay, so this is from my Authentic Biochemistry podcast, which I just put together. All right, so ultimately we are finally at the end. This is, of course, the Continental Divide in Colorado, right at the Wyoming border, a favorite place of mine, Medicine Bow Mountains. There is our email address for the company, info@VerEvMed.com. There's a website. Please go check it out. What VerEvMed does is we are scientists verifying published evidence in medical biosciences. That's what VerEvMed stands for. There is my Facebook page. Again, it's got all of these lectures and lots of different ways to contact me. Here's my individual email address so that you can contact me directly off that, reading that off of this slide. So let's go ahead and close this off. And what I normally do right now is what I'm going to do, like I always should do, is do my sign off. Hopefully this was illuminating to you. And next time we're going to go finally into that last stage where we're going to talk more about the immunoepigenome, and that'll link it up. So we've already given you now sort of my precursor, my preliminary Diaeventontologic of Alzheimer's disease. I want to do that with all the diseases we discussed. And I want you to get an understanding of the fact that this is all a temporal signature on it as well. As time is involved, all of these pathologies move through various levels of discrete interactions. Those discrete interactions can be blocked or proceed at then a new altered rate, depending on the events involved. Not substance ontology, but event ontology, of course. So with that, I will say